So with that, Petra, could you share your screen or I will introduce you while you're doing that? So Petra originally did her PhD in Canberra in Australia, and that was studying plant viruses. After she completed her PhD, she moved to Scotland to work with Carlo Parca's group on virus movement, plasmodesmata, and plant secretory pathways. But in the early 2000s, she moved from plant viruses into the oomycete field and began working on Phytophthora species. And that is the current focus of her research, is understanding the cell biology of plant pathogen interactions with a focus on Phytophthora species that infect both crop plants and trees. So with that, Petra, I look forward to a really exciting presentation from you.
Cool. Hopefully I'm on volume. Yep, right. And are you seeing the proper screen? Because I have two screens on the go, so it's a bit confusing. Right. Okay. So obviously, I can't talk specifically about the details of any one system that you might be using to do your work, or you might use in the future to do your work. So what I'm going to talk about generally is about how to go about making good and useful imaging. So good quality images are those that provide a really clear representation of your results. But it's also worth considering that aesthetically pleasing images increase the impact of your work. So I'll just start with something that basically underpins the value and quality of your images, and obviously any other data that you generate. You may be aware that funding agencies and journals are moving really rapidly towards open science principles and have rules to ensure data integrity. And if you look at journal instructions now, most require authors provide things like raw data on which graphs are based, pictures of whole gels where only parts have been used in figures, and full original images where a cropped or modified version has been shown. The other thing to realize is that if anything in your publication is queried later on, maybe even years later, then the authors or whoever's the responsible author may need to trawl back through all the old results and check them. And very occasionally folk have to remake figures where just an inadvertent mistake has been made. You yourself may have moved on to a different lab, a different field, or even a different career altogether. So please be kind to those you have left behind. So you must ensure that all of your data is safely backed up and accessible either on network servers or the cloud or whatever system your institution has in place in a long-term storage. All of your results have to be accessible. At the very least, everything has to be accessible to your supervisors and the grant holders, as they are answerable to funders and peer reviewers. Your institution and funders also have data policies, which you are contractually obliged to adhere to. So a particular bugbear of mine is annotation. So when you're in the thick of an experiment and you think, oh, of course I know what that is. Of course I'm going to remember exactly what that is, exactly what I did and why I did it. But believe me, give it a year or even less, you will not remember. So make notes, lots of notes, and back them up alongside your images. You need to know, and other people need to know, what each of your images actually represents and what was done. And you must ensure, therefore, that your notes can be understood by others. Because science is a continuum, you are coming after someone else and someone will come after you. It's also essential to preserve raw data for all of your experiments. For images, that means saving the files with whatever metadata is generated by the system you're using. For confocal microscopes that I use mostly, this means saving the files as they're generated by the microscope software. If you're making projections, so you crop the images or you alter them in any way, you need to save your alterations as separate files and never ever overwrite your original raw images. If you're using something like a basic microscope with no digital collection of metadata, then you need to create that metadata yourself by making your notes, recording what lenses and filters, etc. you've been using. So there's something wonderfully tangible about what I love about cell biology is something wonderfully tangible about seeing your experiments, particularly in living cells. And it's really easy to get absorbed in imaging because it's quite pretty and very distracted by it and lose sight of what you're doing. So it's really important to keep sight of your hypothesis and your plan, and to have a plan. Sometimes, obviously, you don't know what you're going to find and haven't developed all that much of a hypothesis yet because you don't know what you're going to get. So preliminary experiments are absolutely invaluable to help shape your project. So a quick look-see can be a very helpful way to find out what kind of hypothesis you're going to need, what kind of experiments you're going to need down the line. Once you have a bit more of an idea of what might be going on, a key question is, what kinds of data are required for robust results? So robust results are best generated by evidence from different experimental approaches. So imaging is very rarely done in isolation. And it's important that experimental setups and controls match up as much as is actually possible and practical. You need to ask yourself if you need qualitative and/or quantitative data. In most cases, both is the answer. And controls, they are the foundation of scientific experimentation. So you need to ask yourself, what are the ideal controls? The ideal controls, of course, might not be available. So then you ask yourself, okay, what's the next best thing that might actually be achievable? It's wise to ask this not just at the beginning, but regularly throughout a project, as your knowledge will change and perhaps you will realize later on that what you thought was the ideal control might not be, or a more ideal control might become available. Specific to when you're using fluorescent fusion proteins or any other kinds of fusion proteins, it's absolutely essential to check that your fusion is actually intact. Otherwise, your results might possibly be completely meaningless. Specifically, when you're imaging, you need to ask yourself and discuss with others what kinds of images will actually be required to generate the data, and what kinds of images will best represent the results. So for localization, I see this a lot. Someone shows a single optical section and says, oh, my localization is this. Now in leaf cells, the vacuole fills the cell and presses everything up against the edges of the cell. So you look at this and mostly it's black because there's no fluorescence in the vacuole. So the whole black area is basically the vacuole. It's not nothing, it's something there, but it doesn't have the fluorescence in it. If you know what you're looking at, you could say, well, you could hazard a guess that, well, this is probably cytoplasmic. If it's a small protein, then it can freely diffuse through the nuclear pore. And so you can see the nuclear plasm, which is pretty much continuous where the cytoplasm is labeled. And you can sort of look at the edges and go, well, you know, it might be cytoplasmic. But you can't really tell. You can see in this image here is also a single optical slice. This is a protein that's localized to the endoplasmic reticulum. You can see the nucleus is dark. It doesn't have any fluorescence in it. So you say, oh, well, okay, of course, that then means that this one's cytoplasmic. But is it really? This cell here has a cytoplasmic protein, but the nucleus is dark. And this could be because the protein is too big, or it could be that it's part of a complex and therefore does not enter the nucleus. And then this cell here doesn't look all that much different from that cell, but this is a protein that's actually localized to the cytoskeleton. So you just can't tell what's going on in these cells from a single optical slice. You need to make a projection. So in leaf cells, for sure, you need to make projections in order to show localization correctly on this sort of scale. With most other kinds of cells, you probably need projection images to show many kinds of localization patterns. So in this cell, we can now see classic features of cytoplasmic fluorescence. The fluorescence is kind of fuzzy, it's not like crisp lines. And there are organelles in negative, which hopefully you can see. As a proof, you could co-localize with a free fluorescent protein, which has got no tags, no fusions at all. as that will just be cytoplasmic. But you could also do other experiments to prove that it's localized, such as cell fractionation, to show that your protein is in the soluble fraction. And this is an important consideration when you're doing images. Of course, a single look at a single sample is not enough to be sure of your localization results. So you need to look at lots of cells in biological and technical replicates to be sure. And then you need to ensure that the image that you use for publication or presentation is an informative and truly representative one. So here's an example for a quantification experiment. So when I looked at co-localization of this particular effector and its putative host target, I noticed that when they were transiently co-expressed, the PP1C fusion was being lost from the nucleolus. Of course, in order to demonstrate this in a robust way, we needed quantification. So what I did was collect many, many, many single optical slice images from biological replications of the co-expression, and then used regions of interest covering the nucleolus and the nucleoplasm and measured the average fluorescence intensities in those. And I did this for combinations with the wild type form of the effector and with a non-interacting mutant effector. And then obviously drew graphs of those fluorescence intensities to show the relocalization was happening. It was being lost from the nucleolus in the presence of the wild type effector and not in the presence of the mutant. From these images, these single optical slice images, some of them were nice enough to actually be then used for presentation purposes. But this isn't always the case. And it might be that you need to collect specific images of the same samples you're using for quantification to make them a little bit nicer so that it's going to be clear, a clearer image in your publication. So what kinds of images are useful to show localization under different circumstances? Well, if you observe that there's some variation in the pattern of localization that you see in different cells, you'll either need to show multiple images of cells to show this variation, or if you can, an image of multiple cells, which kind of gets the point across in one go. If there are also details that aren't clear in a whole cell or multi-cell image, you need to image those at higher magnification, and then you can show them as separate images or as insets like this nucleus to show that detail. Sometimes actually high magnification image of a small area within a cell is really actually the only thing that is useful. Here we're looking at a protein that localizes to the ends of mitochondria, but it also has substantial fluorescence in the cytoplasm in the background, because it's either through overexpression or just because the protein is one of these ones that goes on and off particular sites. The mitochondria are co-stained with mitotracker red, which also has some non-specific background. So a whole cell image, so mitochondria are also extremely small, and there's very, very many of them, and they move. So actually a whole cell image is not really very informative or useful. What I would actually show rather than a single image like this is probably a series of images, and actually also a movie would be really helpful for this particular construct. So the other thing you need to consider is putting objects in context. So if you have a single color of label and then you make a single color image, it might not be very informative. So here we have an array of dots. Well, that's pretty meaningless. If you put a scale bar on, you might say, well, they're quite big dots, but other than that, you really have no idea what they are. If we add a transmission image and also fluorescence from the chloroplasts, you can see that these dots are now associated with infectious hyphae. And so if I say these are Haustoria, you can say, okay, well, I can imagine that they probably are Haustoria. A key basic element that goes into making a nice and a useful image is cell selection and how you choose to actually image that cell. So leaves, for example, even when they appear pretty flat macroscopically, at the microscopic level, they really aren't flat. If you want to make a whole cell image, then it's really wise to select a cell with an appropriate expression level that is sitting fairly flat. So if you consider, so then you won't need to go too deep within your sample in order to make an image of that whole cell area. If you consider when you make a projection, if you're projecting all of the information from the top of the cell and all of the information from the bottom of a cell on top of each other, then that's going to end up really busy. There's just information coming from all over the place. The other thing is that most things in plant cells move, so stuff will be moving around whilst you're going through that cell, and it could end up a real mess. So here's an example. This is a very busy image, and it's basically gone too deep. So it's just, these are mitochondria, they whiz around, there's hundreds of them, and it's just a disaster. If I take only half of the cell, the top half of the cell, and image that, sure, we've still got hundreds of mitochondria, can't really escape those, and they move. That's also a bit of a problem. We could have imaged at a much higher speed to try and sort of capture the Z series before the mitochondria moved too far. They move very fast, so that would have helped a bit, but not completely. But there's also a trade-off going on here. So you can see that there's fluorescence in the ER network as well in these cells. And that's much fainter than the fluorescence in the mitochondria. And if you use a really high speed imaging, then the amount of light collection is much lower, and therefore you will not be able to see the ER. All imaging is a trade-off, really. So an essential bit of skill in performing imaging is obviously ensuring that your images are collected with optimal settings, the best settings that you have on your system for your particular setup. This is where good training and helpful advice are really absolutely invaluable. So in this rather dodgy image, either the Z steps were too large in the series, creating these lines. So this is a plasma membrane localized protein. It should be smooth, and we're getting all of these like topographical lines because the Z steps were too big, and therefore there are gaps in the information. What we also find, though, particularly when we use mounting samples in water, is that sometimes the cells can swell. So that's not to do with the setup, it's just to do with the cell behavior. A somewhat technical point, but something you should inform yourself about, is image handling and preservation of the data within the image. So if you do any actual measurements on an image, you need to be measuring what was actually detected. And that usually means using the image in its raw collected form. If you do post-processing, which removes, adds, or changes pixels in your image, you could lose data, or what's worse is to artificially create it. If you're preparing an image for presentation, you may need to brighten it, crop it, shrink it, make sure it fits the figure, and that it's focused on the results being described in the figure, or some other processing. But you should make sure that your processing is as minimal as it can be, that you're not messing with the data, particularly not creating data that's not there, and that you make a note of what modifications you've done. Something that's particularly pertinent to plant pathology and to defense-related work is that often we're examining whether proteins are responding to change, like biotic or abiotic stress. Often, you don't know whether any individual cell that you're looking at under the microscope is responding to something. And most of the time, it just has to be a numbers game and comparing to controls. You image and measure many, many, many cells, and the overall comparison between the test and the control will indicate whether there is an effect. Pathogen effectors, for example, often target host proteins. Sometimes they change the behavior or localization of those host proteins in a manner that can be observed through imaging. So, for example, that example I showed of the infestans effector O4314 and the PP1C. So if you're looking at such events, particularly in a system in which expression levels are really variable, for like transient expression, for example, you need to take account of the relative levels of the proteins involved. So if I had in one of those cells an absolutely massive amount of PP1C and hardly any effect that I could barely see, would I actually observe any change in the localization of the PP1C? To quantify these sorts of changes that were due to interactions, you can approach the issue in different ways. So in option 1, you could do a test experiment to gauge the relative levels of the proteins that are required to get a detectable effect, and then image and measure cells with a certain set amount settings on your microscope, and only use the cells that have suitable expression levels. Option 2 is just measure everything and change and represent the change as a function of the relative concentrations of each protein. Now, option 1 might sound a little bit arbitrary, but even for option 2, which is kind of the more neutral way of doing it, you have to exercise a wee bit of common sense. So if you are using transient expression, for example, the expression levels can vary really quite dramatically from cell to cell. And there may be cells with no detectable expression of one protein. So is there any point in measuring those? Conversely, there may be cells with really high expression of one protein, which may then damage the cell and cause artifacts, or have levels of fluorescence that actually saturate the detector and actually can't be quantified. So there's no point actually in using those either. The other thing with option 2 is that it might be the ideal method, but it might be more difficult to represent in a way that your audience can easily understand. This is an example where we're looking both at an effector blocking an effect, at both a protein responding to a stimulus and the effector changing that response. So this effector, 03192, blocked the movement of the NAC transcription factor from the ER into the nucleus in response to PAMP stimulus. The issue was that when the NAC transcription factor moved to the nucleus, it was then rapidly degraded. So I needed to use MG132 in order to stabilize it to be able to detect it. So 4 things had to converge in any individual cell that we were looking at. Needed the effector, needed the NAC transcription factor, the PAMP stimulus, and the MG132. I could see the effector because it was being expressed, was actually not tagged, but it was being expressed in a vector that had red peroxisomes also, a red peroxisome tag also being expressed. And I could use that as a proxy for the concentration of the effector. The NAC transcription factor was tagged with GFP. The PAMP stimulus I couldn't see at the time, but you could look at the area a day or so later and it would be starting to die. The MG132, I couldn't see. I just had to mix it very thoroughly with the culture filtrate and hope for the best and image lots of cells. So there are specific complications of working with pathogens and defense proteins. And the golden rule is to always keep the biology in mind. So it's important to learn what normal looks like. So the cell here, it has normal, healthy endoplasmic reticulum. You see a beautiful cortical network. You see the transvacuolar strands and the nuclear membrane is labeled. The other thing to remember is that any ectopic expression is overexpression. But a useful cell for imaging is always a trade-off between the imageability of the cell, because if it's too faint, you just won't get a useful image. But if it's potentially too highly expressed, it will have a higher risk of artifacts. The other thing to remember is that defense-related proteins could be responding to environmental contaminants or stimuli. So if say you have something expressed in a leaf, if your leaf is in the glasshouse or even just in the lab, something could have landed on it from the air, bacteria, fungi, and that defense protein could be responding to those. There's also crosstalk between defense pathways and environmental response pathways. And so in an environmental stimulus, your protein could be responding to that. Of course, pathogens are generally quite damaging. So proteins will change in response to damage, and the structures within the cell will change in response to damage. And so it's important to know whether what you are seeing is a behavior of your protein or your organelle, or is a consequence purely of the disruption that's going on in your cell. So previous slide, I showed healthy ER. This ER is starting to look quite disrupted. There are sort of clumps forming, aggregations forming. Here, it's completely fallen to pieces, and we now have complete disaster. The ER, unfortunately, is quite sensitive and will fall apart nearly at the drop of a hat. So in pathogen infection, this is Phytophthora, the ER falls apart quite readily, and you can't image a cell in that state. So things are quite time sensitive in pathogen interactions, generally speaking. The cytoplasm is less sensitive, but eventually it will also break down and form these clusters of clumps. So it's condensing. You can't use a cell that looks like that. The other thing is your protein showing displaying a behavior, or is it a consequence? So this is an example of a protein that accumulated quite strongly at Hostoria. The image is not great, it's massively oversaturated, but that's to show that the cytoplasm is still in an okay state and the cell hasn't started to die. But you can compare these in massively intense accumulations to this protein, which doesn't specifically accumulate at Hostoria. You can also compare to this cell, where there's an apparent accumulation at Hostoria, but if you look in the rest of the cell, the cytoplasm is in a bit of a state. It's condensed and forming strange clumps of the chloroplasts. So finally, I want to suggest you consider aesthetics and impact. So for gathering data and making sure that you have plenty of replication to ensure that your results are robust, the majority of images don't have to be beautiful or perfect. But for presentation, it really helps to increase the impact of your work if you show high quality and even aesthetically pleasing images. So if the reader of your paper doesn't have to squint and guess at what your image is showing in your figure, they will be a happier reader, or more importantly, reviewer. If when you give your presentation, you can make your audience go, wow, they're more likely to remember your talk. If you can score a cover image of a journal, you will raise the profile of your work. Also, if you have aesthetically pleasing and informative images, they may be really useful for outreach and for advertising the work. And outreach is a really important facet of doing science. So I'll just a quick thank you to all of the people, all of the funding bodies that have funded the work. Thanks to the people who were involved in the science involved in all of the images I've been showing. And I mention also to the people who really taught me my cell biology, Chris, Carl, Ali, and Kat. Thank you for listening. Any questions?
Thank you very much. That was really a very fine talk introducing us to this subject. Well, that's my video button. So I mean, you do produce some stunning images, Petra. I've always enjoyed seeing your talks, or Paul's talks with your images.